Hi, I'm Dr. Nicholas Ashton. I'm the Senior Director of the Fluid Biomarker Program at Banner Sun Health Research Institute in Phoenix, Arizona, and I'm a Target ALS researcher. So my work is predominantly in Alzheimer's disease. My expertise has been developing fluid biomarkers for Alzheimer's disease. But my lab takes the approach of that we're trying to develop tests and assays for pathology, not actually clinical syndromes. So one of those pathologies that's common between ALS and Alzheimer's disease is TDP-43. So we have partnered with many experts within Target ALS and the ALS community to bring our expertise together to see, can we develop fluid biomarkers for TDP-43, which would be relevant for ALS, but also relevant for Alzheimer's disease. My work really is reliant on people having targets of interest that I can go after and my team can go after and say, let's develop an assay for this particular target, which the ALS community have brought forward quite a number of different things. We spent the last decade developing blood tests and cerebrospinal fluid tests for phosphorylated tau and amyloid-beta and neurofilament light in the context of Alzheimer's disease, and we've known what's worked and what hasn't worked, what technologies we've known have worked and what hasn't worked. We can take all that knowledge, package it up, and now apply that with the ALS experts to kind of guide them and help them in what has worked in the Alzheimer's disease space. And we're also learning from them about the biology behind ALS. Cryptic peptides and TDP-43 loss of function is all super interesting and relevant for Alzheimer's disease. So it's really a cross-platform learning curve for both sides. So a biomarker is just a biological measurement. It can be of normal function, but in most of the cases in dementia, we're thinking about loss of function or injury or damage. So this is basically, in our context, measuring proteins in cerebrospinal fluid, which is the fluid that surrounds the brain, or hopefully the golden egg is really to develop in blood, so an easy accessible biofluid. So measuring these proteins in these biofluids, which reflects what's going on in the brain, and these proteins in terms of brain diseases are often in femtomolar and picomolar levels in the blood, so very, very small levels of these proteins, so you need very highly sensitive assays. So we've been very successful, and the ALS community knows this, in neurofilament light. So this is a very well-established fluid biomarker that we can measure in plasma and serum and the cerebrospinal fluid, and we use it to monitor disease. In Alzheimer's disease, we measure phosphorylated tau. That shows if you have tau or amyloid in the brain, and that's used in clinic now to help diagnose patients as well. So a fluid biomarker is essentially, in the context of ALS and dementia, is trying to give us a surrogate marker of injury in the brain. We can't expect every researcher to be able to go through each stages and do the whole pipeline of, in our case, find the target of interest, which is cryptic peptides, do all the basic biology behind that to find those targets, and then get that all the way through to a clinically validated biofluid test. That is decades worth of work and expertise that go beyond just one group. So that's why we have partnered with Len Petrucelli, and that's our Target ALS funded grant, that he has developed antibodies and done a lot of work around the cryptic peptide space, and we're bringing our knowledge in from the Alzheimer's space of developing highly sensitive immunoassays, bringing those together, and what we've seen in the Target ALS is that this is proving to be quite successful. First of all, Target ALS brought us two groups together. That was the first thing. We met at the conference. Target ALS facilitated the introduction and brought these groups together to talk about what our commonalities would be and what our complementary features would be, and then we put in a grant which was funded by Target ALS. Now we've got to the point where we have an immunoassay which we think is working. Yes, Target ALS are going to be able to provide us with the biofluid samples just to prove what is the value of a cryptic peptide loss of function test in ALS. So our job in the lab is to get an immunoassay working which we think is technically validated, which means it's reproducible, it measures what it should be measuring. And now the next phase is that we need to then put this into patient samples to say, is this relevant for a clinician to use? Is this relevant for a drug company to use in their selection and outcomes in clinical trials? So absolutely, from all stages, from the inception of the project all the way to the end, Target ALS has been involved all the way. My perspective on being relatively new to the ALS field is that there is a lot of hope, coming from the scientific perspective as well as the patient care's perspective. So you have targets of interest that I can use my team to develop immunoassays for. We present at Target ALS, we have drugs that seem to show therapeutic response in patients. It's early phases, but data presented has been very compelling. So I'm really optimistic that we can have a suite of biofluid tests that can help us untackle the heterogeneity around ALS. This is where we come from Alzheimer's disease. Alzheimer's disease, we know now, just isn't amyloid and tau, these two key proteins. It does have TDP-43 and it does have alpha-synuclein. This is a very, very heterogeneous disease, and we see commonalities even with ALS. So I'm very optimistic that we can develop something very specific for ALS, very important for this community, but that can actually be used more globally. At the moment, we are measuring cryptic peptides in cerebrospinal fluid. So this is often a turn-off for research and for patients and for clinicians, that this is an invasive biofluid to collect. So our aim and our hope, and I am optimistic that we'll be able to do the same things from a biofluid, from blood. So that means that it's easier for the patient to give the sample. It means you can monitor that patient more regularly because you can give weekly, monthly, annually biofluids. The way that we've progressed through Alzheimer's disease to go from CSF to blood so quickly, knowing the target of interest, I'm confident that we'll be able to do that with the biomarkers that we're measuring in cerebrospinal fluid for ALS. The whole idea around cryptic peptides is that TDP-43 forms within the neuron and causes a loss of function. And what that produces is a peptide that is not usually produced by the body, that leaks out of the cell into the biofluids. Now, sometimes we call this in frame and not in frame. And if it's in frame, it means it can get into the biofluids and gives our group a chance to be able to detect this in cerebrospinal fluid and hopefully in blood in the future. So in theory, the principle is that if TDP-43 is present in the neuron and in the brain, that this peptide is produced specifically because of that. And if you can detect it, then that means that the pathology is there. My name is Dr. Nicholas Ashton. I'm the Senior Director of the Fluid Biomarker Program at Banner Alzheimer's Institute, and I'm building a world where everyone lives.